Okay, today we'll be discussing paper 2 MCQ's biology major 2023 variant 21. So let's start. First question: what are characteristics of all organisms? To tackle this question, we need to know what are the characteristics that all organisms have. And as we can see over here, the characteristics that are followed by all organisms are movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. And this can be memorized by by the acronym of Miss Gran. Now, so going back to our question, as we can see, what are characteristics of all organisms? Egestion, and excretion? No, and that is because this is wrong. Digestion is followed by mostly like animals, which is the way they excrete their waste materials like feces, urea, urine, and thus. An excretion, yes. Digestion, no. Nutrition, so these options are wrong. Excretion and nutrition, yes. Excretion and photosynthesis, no, because photosynthesis only occurs in plants that do have chloroplasts. So the answer is C. Second question: the scientific name for the golden eagle is Aquila crisatus. What is the genus of the golden eagle? Again, going back, as we can see, this question talks about the binomial naming system, which is an internationally agreed system in which the scientific name of an organism is made up of two parts showing the genus and species. As we can see over here, the scientific name of a wolf is Canis lupus, and to write the scientific name, so as first of all we need to make sure the whole name is in italics and the genus, which is the first name, is capitalized. So going back to our question, the answer is going to be A, Aquila, as is the name of the genus. Question 3: which row shows the structures that are present in both root hair cells and palisade mesophyll cells? Again, going back to this question, let's display both cells. Root hair cell and a palisade mesophyll cell. So as we can see, both of them do have a cell wall, and that is because they're both plant cells, and plant cells have cell walls, so this should be right. Secondly, chloroplasts. Palisade mesophyll cells have chloroplasts because they are the main site of photosynthesis in the leaf, whereas the root hair cell is present in the roots and it does not need to carry out photosynthesis as it's not its function. So it does not have any chloroplasts. Cytoplasm, yes, it's present in both, and that is because they are all living. Cytoplasm is present in all cells of living organisms or just organisms. A vacuole, yes, all plant cells have a vacuole, which contains cell sap, which is like a mixture of salts, water, and thus. So, looking back at our question, we can clearly see that the answer is C. Okay, The micrograph shows a plant cell. The length of line XY is 90 millimeters. The actual length is 30 micrometers. What is the magnification of the micrograph? Okay, so in this question, we can look at this triangle where I is the image size, A is the actual size, and M is the magnification. And since we need the magnification, we're gonna divide image size by actual size. And by looking at our question, we can see that not all units are same, as the actual length of the cell is 30 micrometers. And when solving magnification questions, we need to make sure that all units are same, and we're gonna change the micrometer into millimeter, because we should always change it to millimeter. Anyways, so the 30 micrometer we're gonna change it, so 30 micrometer to millimeter. We're gonna divide 30 by 1000, and that value is what we're going to use in this question. So I'm going to take out my calculator so I could, yes, we're here. So let's divide. So 30 micrometer divided by 1000, 0.03. So I'm going to take the image size, which is 90, and divide that by 0.03, which is the actual size, to get an answer of 3000. So the answer is going to be B. So now let's cross-check our answers with the marking scheme. So we got C, A, C, B. So C, A, C, B. Right. Now moving on to the next question. The diagram shows an experiment demonstrating osmosis using a dialysis tubing bag. After 30 minutes, the level of the liquid in the glass tube goes 1, because the water had a 2 water potential than the sugar solution. Which words correctly complete the statement? So what is osmosis? So let's go over here. So osmosis as the net movement of water molecules from a region of higher water potential to a region of lower water potential, which is a concentrated solution, through a partially permeable membrane. This dialysis tubing acts as the membrane, and here we have a glass tube where the sugar solution, where it's more concentrated, which means lower water potential. In this case, since it's more concentrated solution, it's a lower water potential, and this water has the greater water potential. And in osmosis, water molecules move from a region of higher water potential to a region of lower water potential. So the net water movement is going to be from the water into the dialysis tubing. Thus, the level of the solution in the glass tube is going to rise. So let's go back to our question. After 30 minutes, the level in the glass tube goes up because the water had a higher water potential than the sugar solution. So the answer is going to be D. Okay, moving to the next question. Since this is alone in one page, we're going to just cross-check it too. 5 is D. So moving on to the next question, question 6. The diagram shows the movement of glucose molecules across a cell membrane. Which statement describes molecule T? As we can see over here, this is the process. Since it's glucose molecules, of course, and energy is required, it's the process of active transport, and there is a membrane. So to check this, as we can see, active transport is the movement of particles through a cell membrane from a region of lower concentration to a region of higher concentration against the concentration gradient using energy from respiration. And what's actually happening here is the protein carriers move the molecules or ions, and in this case the glucose molecules, across the membrane during active transport. So obviously this protein T, it's a protein molecule because they are protein carriers, and they are used to transport glucose against the concentration gradient since active transport is happening. So the answer is going to be D. Question 7. The diagram shows part of a protein molecule. What does X represent? Since this is a protein molecule, of course we need to see the building blocks of a protein molecule. To check this, as we can see, we know, and this is from the syllabus, state that large molecules are made from smaller molecules limited to starch, gluten. Okay, so B, proteins from amino acids, and there are like about 20 different amino acids with different sequences forming different types of proteins. So the answer is going to be A. Question 8. Of DNA involves two strands called together to form a double helix. Which pairing of bases between the two strands is correct? So A and G, B, A and T, and C and A, D, C and T. So to tackle this question again, let's go back to check out the pairings from the syllabus. Describe the structure of a DNA molecule. Its two strands coiled together to form a double helix, as we can see here. Each strand contains chemicals called bases. Bonds between pairs of bases hold the strands together, and the bases always pair up in the same way, at A with T and C with G, adenine with thymine, cytosine with guanine. So to know the right answer, let's go back. Okay, so it's going to be B. Okay, let's check these again. Question 6 is D. Okay, question 7 is A and 8 is B. So A, B. Okay, now moving on to the next question. What is true of all enzymes? Okay, so first of all, let's talk about enzymes. Enzymes are proteins that are involved in all metabolic reactions where they function as biological catalysts. And of course, catalysts are substances that increase the rate of a chemical reaction and is not used or changed by the reaction itself. So let's read our question. What is true? They are sugars. They are most effective at pH 7. Enzymes are proteins, thus they are not sugars. So X, the first one is wrong. They are most effective at pH 7. So first of all, we need to know how enzymes are affected. Enzymes are affected by pH and temperature. So if the optimum pH or optimum temperature, where the enzyme acts as in its most, is exceeded, the enzyme gets denatured where the active site no longer works. So in this case, they said that most enzymes are effective at pH 7, which means that all enzymes need to work at the optimum pH of pH 7. But the thing is, we have pepsin, which is an enzyme that works in the stomach in acidic conditions, which means, and that's just one example, which means that not all enzymes are most effective at pH 7. So the answer is going to be D. Question 10, which statement about human enzymes is correct when they are at temperatures above 80 degrees Celsius? The enzyme molecules are denatured, the shape of the active site is stable, more enzyme substrate complexes form, more product is formed by the enzyme. So check this question. Investigate and describe the effects of changes in temperature and pH on enzyme activity with reference to optimum temperature and denaturation. So when the optimum temperature or optimum pH of an enzyme is exceeded, then the active site of an enzyme gets denatured. Thus, it is no longer effective and does not work. So in this case, as we can see, the answer is going to be A, because 80 degrees Celsius is way above the internal temperatures of a human being. So the shape of the active site is stable. The second one is wrong, of course, because the active site gets denatured. More enzyme substrate complexes form, no, and that is because again the active site is denatured, and thus it does not form any enzyme substrate complexes and is deemed useless. More product is formed by the enzyme, no, and that is because, of course, as we said, it's denatured and it does not work. Question 11. An experiment is set up to investigate gas exchange in aquatic plants. The hydrogen carbonate indicator solution is orange at the start. Which color is it after 3 hours? Since light is present, and as we can see, all the conditions for the plant to photosynthesize are present, available, so photosynthesis is going to occur, and this carbon dioxide is oxygen is oxygen is going to be formed, and carbon dioxide is going to be taken in by the plants. So first of all, what's the hydrogen carbonate indicator? It's an indicator for the carbon dioxide levels. When the level of carbon dioxide is normal, as in the rate of respiration equals the rate of photosynthesis, then the hydrogen carbonate indicator will be red. If it's more, it becomes yellow, and if it's way less, the levels of carbon dioxide, it becomes purple. And since what's happening is photosynthesis, then oxygen is going to be released and carbon dioxide is going to be taken in, which means that the carbon dioxide levels are going to become lower in the water solution, so it's going to turn purple. So the answer is C. Let's recheck our answers with the marking scheme. 9 DAC. Okay, so 9 DAC. Next question number 12. Certain factors are needed for photosynthesis. If a factor is in short supply, it limits the rate of photosynthesis. Which of these could be limiting factors? Carbon dioxide, 2. Oxygen, 3. Light intensity. Okay, so we need to check what are the factors that the rate of photosynthesis depends on. And those are light intensity, carbon dioxide, and temperature only. And thus, what could be the things that can be a limiting factor are carbon dioxide and light intensity. So the answer is going to be B. Oxygen is not included, and that is because oxygen is a waste product and not a reactant. Second question, question number 13. The hemoglobin concentration in the blood of a person is 80 grams per decimeter cube. The accepted normal concentration is 120 grams per decimeter cube or above. Which substance may be lacking in their diet? So, hemoglobin is made from iron. So before that, let's check the vitamin and minerals required. So as we can see, iron over here is needed to make the hemoglobin pigment in red blood cells that transports oxygen. And so if the hemoglobin levels are low, that means the iron levels are low. So as you can see in this question, which substance may be lacking in their diet? It's iron. Question 14. Which statement about physical digestion is correct? Okay, physical digestion. As you can see, physical digestion is the breakdown of food into smaller pieces without chemical change to the food molecules. And physical digestion increases the surface area of food for the action of enzymes in chemical digestion. So basically it's just tearing down of substances, the same as tearing down a piece of paper. That can be considered as physical digestion, but in physical digestion it occurs in like way smaller molecules. So which statement about physical digestion is correct? It increases the surface area of food. Yes. Involves enzymes? No. Then it will be chemical. It takes place in the mouth only? No. It does take place in other places such as the duodenum, the stomach, and many more. It produces smaller molecules. No, they're the same molecules, but they're just increased in the surface area. So 14A, because the last one is wrong, and that is because the molecules themselves are not touched. If smaller molecules are produced, then it's going to be chemical digestion. Okay, 15. Which statement describes the effect of atmospheric humidity on the rate of transpiration? Okay, in high humidity, the transpiration rate is high because there's low diffusion and okay, so this question talks about transpiration. Okay, so in this case we have temperature, wind speed, and humidity. When temperature is more, the rate of transpiration is also more. And that is because particles have more kinetic energy, water particles have more kinetic energy, thus increasing the rate of evaporation, and thus increasing the rate of transpiration. As in wind, if the wind speed is more, the greater the transpiration rate. And that is because this increases the concentration gradients between the air and the leaf where transpiration occurs. So for example, here we have the evaporated particles. In this case, if we have a greater wind speed, each time the particles are transpired from the reed, they're gonna be blown away, and thus the concentration of grain is gonna be maintained, and it actually makes sure that these particles do not condense back into the leaf. And finally, humidity. When humidity is high, transpiration rate is low, and that is because it lowers the concentration gradient. As we can see, if there is no, let's cut out this wind, we have here the particles. First of all, there is a greater possibility that the particles are gonna go back, and the concentration gradient is gonna be low, reducing the transpiration rate. So going back, in high humidity, the transpiration rate is high. No, it's low. In high humidity, the transpiration rate is low. Yes, because there is a rapid diffusion of water vapor through stomata. No, it has a low diffusion. In low humidity, the transpiration rate is high. Yes, because there is a rapid diffusion of water vapor through stomata, and that is true. So the answer is C. Okay, so 12 BDAC. 12 BDAC. Okay, moving on. Question number 16. Sucrose and amino acids move around the plant from sources to sinks. Which row shows the sources and sinks? Okay, so this question talks about translocation, which is the movement of sucrose, which is carried instead of glucose because it's a larger molecule, through by the phloem around the plant. So what are sources and what are sinks? Sources is where the substance is produced, and sink is where it's used or stored. So in this case, we're talking about the root cortex cells, the xylem vessels, and the palisade mesophyll cells. The root cortex cells can actually be considered both, and that is because they're going to be considered sink, and that is because they cannot make their own food. Of course, they don't have any chloroplasts, so they can be considered as a sink, where they are stored or used up, and they can be considered as a source, and this can be in the growth period, and that is because the leaves are not going to be able to make their own food, and thus they're going to be using up the food stored in the roots. So root cortex cells can be both. Xylem vessels, on the other hand, are basically tubes to carry water and some minerals, and it's just a means of transportation, so it can neither be a source nor a sink. And for the last one, palisade mesophyll cells. Palisade mesophyll cells are the site of photosynthesis, and thus they make their own food, and they can be considered a source, not a sink, because they do not store food. So going back to our question, we can see that the answer is A. Which statements describe how the structures in the circulatory system function? So how do they function? A muscular pump to push blood into vessels, and that's the ventricle. Valves to ensure one-way flow of blood. Yes. Veins to take blood away from the heart. No. Arteries take blood away from the heart. Vessels to return blood to the heart, yes, because and the vessels that return blood to the heart are the veins. Okay, so before that, as we can see here, this is a circulatory system, which is a system of blood vessels with a pump, which is the ventricles over here, those two, right and left ventricle, and valves, which are those, the atrioventricular valves and the semi-lunar valves, which ensure the one-way flow of blood. And then we have the veins and arteries. Veins take blood towards the heart, and arteries away from the heart. And moreover, we have the vessels. Blood vessels include capillaries, veins, and arteries. And the vessels that take blood towards the heart is the vein. And so, in this case, our answer is going to be one, 2, and 4, B, because veins take blood towards the heart, not away from the heart. 17B. The diagram shows a section through the heart. Which part is the septum? So, the septum is going to be this part. This is the whole thing. Or basically just the middle part, this part. And this septum, this muscular wall, separates the deoxygenated blood from the oxygenated blood. So the answer is going to be C. What to keep in mind is that the septum is in the middle part and not on the sides, and these are just the ventricle walls and whatever. And the reason is, as we said, it separates deoxygenated blood from oxygenated blood. Now let's check our answers. 16A, B, C. 16A, B, and C. Okay, moving on to the next question. The cholera bacterium toxin causes dehydration and loss of salts from the blood in humans. Which statement explains the reason for this? And to figure this out, we're gonna look through the process. Okay, so first of all, the cholera bacterium goes into the moves through the small intestine, and then it starts secreting a toxin which causes the small intestine walls to secrete chloride ions, thus lowering the water potential in the small intestine itself. And thus osmosis occurs, and water from the small water moves from high water potential, which is in the small intestinal cells, moves into the small intestine, and thus resulting in dehydration and loss of ions, which are the chloride ions from the blood, and the water which occurs due to the osmotic movement. So let's go back. So first of all, A, chloride ions are secreted into the small intestine where they increase the water potential. No, they decrease the water potential. Chloride ions are secreted into the small intestine, which causes water to move into the intestine by osmosis. Yes. Chloride ions are secreted into the small intestine, which causes water to move out of the intestine by osmosis. No, it's into. Because in this case, out of the small, it means like this into the cells, but no, it's the opposite. Chloride ions are secreted into the small intestine, which causes the water potential of the blood to decrease. No, it, it does not cause the water potential of the blood. It causes the water potential inside the small intestine to decrease. So the answer is B. During inspiration, the processes less take place. Volume of the thorax increases, air rushes into the lungs, pressure in the thorax decreases, external intercostal muscles contract, diaphragm moves down, ribs move upwards and outwards. What is the correct sequence for these processes? Again, to tackle this question, we're going to see what happens in each case. So first of all, the inhalation, when we inhale or inspire, it's the same thing. First of all, the rib cage expands and the external intercostal muscles contract. Internal intercostal muscles relax, they are antagonistic. The diaphragm contracts because it's originally dome shaped, but when we inhale, this dome shape flattens, thus it contracts, and the thoracic volume increases, of course, and the thoracic pressure increases. No way, the pressure decreases because the volume and pressure are inversely proportional, and air is taken in. So, and the exact opposite happens in exhalation. Relax and then contract and then relaxes, and thoracic volume decreases and the pressure increases. So going back, so first of all, the external intercostal muscles contract, one, and then the volume, like this, and then the diaphragm moves down, ribs move upwards and outwards, as in they expand, so that's 2, and 3, the volume of the thorax increases, the pressure in the thorax decreases, and the air rushes into the lungs. So it's STPRQ is B. So B and B, 19B, 20B. And thus we are done.